It depends on you.
How do you do it?
English a bit slower. Okay.
No, Fabio Pardi is responsible of the Projet Intégré ADN Génomes, so Génomes nowadays, and he's going to present the activities. And after that, Stéphane Guindon, I hope I pronounced correctly, is going to present Nouvelle approche en statistiques computationnelles pour mieux comprendre l'évolution moléculaire.
Okay, so English or French?
As you like.
Who in Italian? Yeah, I can do it in Italian. No, that's not possible. Okay, English. So I'm Fabio Pardi, and I'm one of the two people who coordinate the PI Genomes. First of all, let me remind you what a genome is. It's the set of information that is contained in a cell and determines which molecules are produced by the cell and at what time. So it does determine the functioning of the cell. And the goals in our PI are, first of all, to have a better understanding of the organization, function, and evolution of genomes, then to have methods to treat and analyze the data that comes out of genomic and post-genomic experimental techniques. And we would like also to provide tools that can be actually used by people who work in healthcare, in agronomy, in the environment, in evolution as well, and in biodiversity. So we have quite a lot of research themes around which our work revolves. But let me tell you, first of all, that we have our own butterfly in this PI, because we have the techniques that we use come from mathematics, from biophysics, from biostatistics, and bioinformatics. And you'll see in the different axes where this is. Yeah. So one of the research themes that is very important is the analysis of high-throughput sequencing data. As you probably have heard, there are lots of new techniques for sequencing DNA, but these techniques need tools to analyze the outputs of these techniques. And I won't speak of all the things that we have to do with these sequences, but basically we need to develop new algorithms that are based on string analysis and string processing and graph theory and other discrete mathematics methods. Then there is the field of functional genomics, where we are involved in the development of methods, models, algorithms, and software to study how the components of the genomes and the products of the genomes interact to determine the functioning of the cell. You heard yesterday about the flagship project GEM, and the GEM project was born out of our work in this context. Then we have evolutionary genomics, which is very strong in Montpellier. And here we would like to help in the evolutionary analysis. So basically we take genomes coming from different either species or different individuals from the same species, and try and understand how these genomes are related and what led to the genomes that try and understand the evolution that led to the current genomes. So here we speak of phylogenetics, phylogenomics, and population genetics. And then, of course, we also have the biophysical and mathematical modeling of genomes. For example, one very important problem is to understand the spatial organization of genomes inside cells and how this organization changes with time. So the movement of genomes in cells. Okay, so all these research themes involve collaborations with people who work outside NUMEV, so in biology and health labs, for example. We'll see afterwards some collaborations with some labs here, and also with labs that are more oriented towards agronomy, biodiversity, and environment. So let me tell you something about some of the research that has been done in the last year, on the last 2 years maybe. So actually this is 3 years, because this is the thesis of Sylvain Poulicani, who's defending, I don't know if you can say this in English, but who's having his PhD Viva, PhD examination today. So not in this building, but not very far from here. And his work was on the link between the 3D proximity between pieces of the genomes and the evolution of genomes. So nowadays we have techniques that produce this sort of data, matrices, that tell us which part of a genomes are in contact with which other parts. Like here, for example, these are all in contact, in 3D, obviously. And this can be very useful for the study of evolution, because when chromosomes are very close or in contact, a hypothesis is that they are more likely to exchange pieces, in fact undergo what's known as recombination. And when you have recombination, you form new chromosomes that are basically the mosaic of old chromosomes. So here we have a chromosome that is red and blue. And so he had to test if there is this relationship between, if there is actually this data can predict something about evolution, and actually it does. Okay, another work that we are funding, and it's just started, it's the development of a platform for phylogenomic analysis. Basically, in Montpellier, a lot of software has been developed in this area, and an important problem is to integrate this software and to make it easily accessible to people who are outside Montpellier. And this is exactly what Morgane Soulier is doing. Third, this is a project about the plasticity of bacteriophage genomes. Bacteriophages are viruses that infect bacteria, and this was possible thanks to the visit of Anne Bergeron, who's a professor in Montreal, and this led to the maturation, let's say, of a software called Alpha, which allows the genomes of different strains of the same bacteriophage to be compared. And you have to know that bacteriophages have a very modular structure, which means that it's sometimes impossible to align precisely two parts from two different strains. So you have to know which parts are alignable, and this is exactly what this tool does. It says that this part is alignable among all the strains. There are 4 strains here, and then you have 2 strains that are alignable together, and the other 2 are alignable together, and then so on and so on. So it produces a graph like this, and this is very useful. And this is something that we also discussed last year. So this is a project about the segregation of bacterial genomes. When a cell undergoes division, the 2 copies of the genome must go in the 2 different cells. And so what Jean-Charles Walter did was to model how this segregation happens using tools from physics, and the models that they've developed actually describe very well, I would say, what happens in reality. Another fact that is very nice for us is that Olivier Gasquel, who worked a long time in LIRMM and who was very involved in NUMEV, received the prize of the Grand Prix in INRIA Académie des Sciences in 2017. Okay, and let me just end this with a word about the scientific animation conferences and the pedagogic side of our PI. So the PI Genomes funds helps the organization of several conferences. Two of these are SMPGD, which is about the biostatistics of high throughput sequencing, and actually no, high throughput biology, so every kind of high throughput data. And mathematical and computational evolutionary biology, which is about methods from maths and computing to study evolution. And these are conferences that have become quite important internationally in the last few years. And this is about the participation of the master students to Jobim, which you already heard from Anna-Sophie yesterday. So with this, I'm ready to leave the word to Stéphane, whose project I didn't talk about, so he will talk about this a bit longer.
Thank you very much, Fabio. Some questions? Okay. So. Ça marche. Quand il est sorti. C'était pour vérifier que ce soit. Toi, un BGA?
Ouais. Okay, so sorry for starting late. So I'm going to tell you about two projects, or actually 3 projects I've been involved in a relatively recent past. And the main goal here I had was to use genetic sequences and turn those genetic sequences into documents that can be used to date past evolutionary events. And I'm going to try and convince you that there are interesting statistical problems that come with the sort of questions I'm interested in. And I've overcome some of those statistical issues. And I'm now in a position to go a bit further than just developing statistical models of molecular evolution and really use those models to really start make interesting biological inference about the biological processes taking place at a really fine scale of evolution. So this is the project that Marie Suez, who's a postdoc funded by NUMEV, is currently working on. And that's the third part of my talk. So once again, the main goal here is to give you a brief intuition about one quite simple thing, which is to use molecules collected across different species, for instance. So you have one gene encoding for a given protein, and you have the sequence of that gene collected in different species. And from the differences you observe among those different genetic sequences, you want to turn those differences into calendar time units. So you want to be able to date past evolutionary events and transform the differences you see, so elapsed time in terms of mutations in DNA sequences, into calendar times. And the basic motivation is that the more divergent two species are in terms of the time since they diverged, the more different their genetic sequences will be. So this idea of using molecules as documents to date past evolutionary events is not new. So it's based on this idea called the molecular clock, so using molecules as molecular clocks. And that was used about 50 years ago in a landmark study by Alan Wilson that used DNA sequences from apes and from humans, and he was able to reassess the time elapsed since the divergence between apes and humans. So that was the first very noticeable and very nice use of genetic sequences, and once again transform amounts of differences between molecules into actual calendar time units. So how does this thing work? Well, so you start off with DNA sequences here at the bottom. So you have two species here, A and B, and you have collected their DNA sequences. So at the same position along the genome, you have the sequences for those two species. And you observe some differences here between those two sequences. Here, this C for species A is a G here at the same position for species B, and so on and so forth. And here, what from the observed differences between those two sequences, what you're able to do is you're able to estimate the length of that path here that separates those two genetic sequences. And at the top here of that path here, you have the most recent common ancestor of your species A and B. So you know, you can estimate the number of mutations here. So L1 here is an expected number of mutation or substitutions that occur at every position here along the sequence, along that branch that separates the most recent common ancestor of A and B and species A. Right, and you have the same on the other sides. So here, the time is expressed in terms of units of mutation. L1 are expressed in terms of expected number of mutations. All right. So if you want to turn this molecular time into a calendar time, what you need to do, of course, is some more data. Right? You can't just do that by just looking at your two DNA sequences. What you need is something more. And what you can, if you're lucky enough, and if you know the rate at which substitutions, the rate at which mutations along those two branches occur, so you assume here that you have a rate that is common to those two branches, then what you can do is turn those length estimates into calendar time units, all right? Because those length estimates here are the product of the rate of substitution by the time elapsed, the calendar time elapsed along those branches. So you're able to invert your equality here and estimate the time from the branch length. All right. So that's only if you know what the mutation rate is. Now, if you don't have this information, you may have some other information, because usually we don't know what this mu, what the rate at which mutations accumulate is. But what we can have as extra data is fossil information. So here you have fossil information, and this fossil here, if you're lucky enough, then this fossil belongs to the most recent common ancestor of your two species here. So what, and you know how far you had to dig in order to find this fossil, so you have a rough idea of when this fossil exists. And this when is expressed in calendar time units. So you know the time elapsed here along that path, right? So if you know the time here, what you're going to be able to do is, so you have the time here for this node, and maybe a question you want to solve is about the calendar time for this particular event where species B and C diverged, all right? So how can you use fossil information in order to date that? Well, it's fairly simple. Once again, you're able to estimate amounts of mutations along those two branches, and you know that this total amount here, L1, corresponds to a time T1. And you were able to estimate alpha here using your molecular sequences. So here, once again, you can turn your molecular estimates into calendar time units estimates, right? So that seems fairly straightforward. However, there are some complications so that's the standard model. So that's if everything goes well, you have the same rate of mutations here everywhere in the tree, and moreover, you have no uncertainty around the placement of your fossil. You know for sure that this fossil calibrates this node here. However, in practice, things are not so sweet. And in fact, you can't generally assume that the rate of mutation is the same everywhere in the tree. And instead, what you have to assume is that each branch here has its own rate multiplier here, right? R2, R1, R3 are rate multipliers. So they come to multiply this average rate mu, right? And they tell you, for instance, that this rate here is greater than the rate that applies on that branch. So this species here evolved faster than that lineage here. All right? So you have to accommodate for this heterogeneity, this variability of rates of evolution along the phylogeny. And another difficulty here is that you're not always 100% sure of the placement of your fossil. Maybe the fossil calibrates this node here, but, well, given the morphological data of your fossil here, maybe there's a chance that it could calibrate this node as well. All right, so there are those two difficulties that we have to overcome, and that's the two questions I've been working on for quite a long time now. So how do we solve these two problems? Well, we solve them by presenting the problem we're interested in into a nice and sound statistical framework. So in this framework, what we have, of course, is data. So you have your genetic sequences for the different species, and on top of that, you have some fossil information. Now, the model we are interested in, the phylogenetic model, has different parameters. We have the tree topology, tau. We have the height of the different nodes in the tree. That's a vector of node ages. We have the mutation rates, so we have this overall mutation rate, mu, and we have this branch-specific mutation rate. And we have another parameter, which is I'm going to tell you about those calibration constraints. They come from fossil data, and we'll see that we have to consider calibration constraints as an actual parameter in the model. Right? And the way we proceed with this sort of model is we build this model in sort of hierarchical fashion. From the genetic sequences only, so we leave aside the fossil data for now. From the genetic data only, we can estimate a tree topology and a vector of edge length. So we are able to estimate the phylogeny, all right? So how species group with each other and the amount of evolution on each lineage in the phylogeny, right? So that's the first level in the hierarchy. The second level is when you add fossil data. I've already told you about this. When we add fossil data, what we are able to do is we are able to disentangle the rates at which mutation occur and the actual calendar times. All right, those branch lengths here, they can be decoupled and they can be expressed as the product of those two parameters that can be identified from the genetic and fossil data. And in order to make the inference, so we rely on the Bayesian approach, and we have to express the joint posterior density of our model parameters. So once again, the tree topology, the branch length in terms of time units, the rates on each branch, the overall rate, and the calibration constraints given genetic and fossil data. All right, and this posterior density is expressed in terms of the probability of observing the sequences given the model, the phylogenetic model. So I'm going to tell you about that term very shortly. And then there's this term, the rates along each branch given the phylogeny. And then there's the rest that I'm going to tell you about later on. It's not a prior, okay? It's not a prior because here we have some data here coming from the fossils. So this is a prior here on the mutation rate, but this is not a prior, so we have to be careful with that term. But for now, I'm going to tell you about how we calculate this term and then that term, right? How we calculate this term, so the probability of the genetic sequences given the phylogeny, is something fairly well known. So once again, the model is made of a discrete component, the tree topology. So the topology describes how species group together. So for instance here, if we have 4 species, we have A and B together and C and D on the other side. Another topology would be to have A and C together and B and D on the other side. So that's one component of the model. Another component is made of the branch length. So that's a vector of number of mutations. And in population genetics, if you're interested in population genetics, we have some more continuous parameters that I won't tell you about today. So the phylogenetic model, So we want to calculate the probability of observing a given site of our genetic sequences. So here we have one site, so we have the alignment here, 4 species, and we look at the observed characters at that site, all right? So this is the characters we observe at the tips. And if we want to be able to calculate the probability of the whole alignment, what we do is we introduce an independence assumption, meaning that we consider that this site is independent from the other sites, and we're going to be able to multiply the probabilities that we've obtained for each site together to get the probability of the whole alignment. So that's an algorithm that's well known. It was proposed in the 80s by Felsenstein, and basically it's fairly efficient, and using this algorithm we can go up to 5,000 or 10,000, maybe now 100,000 or so sequences, all right? And what's important to keep in mind here is that if we want to be able to calculate the probability of observing the states here at the tips of the phylogeny given the topology and the branch length, this calculation relies on our ability to calculate those transition probabilities. What is the probability to go from an A here at this node to a C or a G at that node, and then from a C to a G along that branch, all right? So the core calculation here of the likelihood relies on those transition probabilities. So this is fairly important to keep that in mind. So we are done with this term here. And now we're going to move on to that term, and that's where the first hurdle comes into play here. And that's the first question I've worked on. So here we want to be able to calculate the probabilities of the different rates along the different branches here, given the tree topology, tau, and given the times of the different nodes here. And it's not straightforward to propose a model here to calculate those average, this density here that describes so the distribution of the rates along the branches given the topology and the heights of the different nodes. What is easy to do instead, what is fairly natural to do, is we can propose a model that describes how the rates fluctuate along the branch, right? So here we have the rates at a particular point, here x1, and the rate at the bottom of the branch x2, and we want to be able to describe how the rates fluctuate randomly along this branch, and what we can and once we have a model like that, we can make some we can calculate the distribution of the average rate here on that branch. So this is exactly what I've done here. So here the model I've proposed is not new. That was already a model that was used before, and what we do is x1 and x2 they fluctuate according to some sort of Brownian motion, and more specifically, the logarithm of the rates of evolution here at the end of the branch is normally distributed around the logarithm of the rates at the beginning of the branch, right? So we have a geometric Brownian motion. The log of the rates evolve according to a Brownian motion, all right? And the standard approach that was used before I suggested that we should do otherwise was just, all right, to have this sort of formula. Here you have the log of the rates at the beginning and the log of the rates at the end. What we do is simply take the average of those two rates in order to work out what the average along the branch is, all right? But of course, if you assume that there's a Brownian motion going on along the branch, then what you really need to do in order to work out what the average rate is to solve this sort of integral here. And this integral is fairly tricky because it's a stochastic integral, right? X here is governed by a stochastic process, all right? The log of x is a geometric Brownian process. So here we have to integrate over an infinite number of rate trajectories, all right? In order to work out what the distribution of the average along the branch is. All right, so this is what I've done here. So using the standard approach, what you can do here is, in order to work out what this matrix is, and this matrix tells you the probabilities of changing from one nucleotide to another along the branch. Well, this matrix here is simply the exponential of the rate at which mutation occur by the time elapsed and the average rate. And as we've seen before, what we want to do here, instead of considering that this is a fixed quantity, we want to acknowledge the fact that this is not a fixed quantity. This is a random quantity. And therefore, we have this stochastic integral that we want to be able to deal with. So that looks fairly daunting to solve this sort of integral, but okay. But what we've done, what we've looked at here is what is the distribution of the average rates when you have those fluctuations along the different branches here. And it turns out that this distribution here is a gamma distribution, and that's very helpful because if this is a gamma density, then you can actually, there are solutions to this integral. So we were really lucky that an analytical solution for this integral existed. All right? So that gave us two articles. So one here was published in Systematic Biology, where we make this approximation that the average rate is gamma distributed, and then we solve the integral. And later on, I've been contacted by Nicolas Privot, who's a statistician, a probabilist interested in financial statistics. And he told me, okay, oh, Stéphane, I can do way better than you did. I can actually solve this stochastic integral without relying on an approximation. So that's what he did. And we were able to show that the approximation I proposed wasn't that bad anyway. So that was nice to have Nicolas involved in this project. All right, so I'm going to skip this second question here on using fossil data in order to date past evolutionary events. I just want to mention here that that there's an interplay between statistics and computational science, and I've benefited a lot from the work of statisticians like Murray here, and basically they've described a statistical method to solve the problem I was interested in fairly recently, and I was able to translate their solution into the context of molecular dating, and that was really helpful to interact with statisticians around those questions. All right, now I'm just going to take 2 minutes to tell you about the work that Marie Suez is doing right now. So we have now a nice model to describe how mutations accumulate along the phylogeny. And what we want to be able to do is we want to be able to try and tell what's happening at individual positions along the sequences. What can we tell about the mutation processes at individual positions along the sequences? Right? So once again, what we have, the data we have are the genetic sequences at the tips, and we know that some mutations occur along the phylogeny to give rise to the observed data at the tips. And there's of course an infinite number of scenarios of mutation that can lead to the observed patterns at the tips. All right, so we have to integrate over all those mutation histories, and what we want to be able to do is integrate over all those scenarios, so the different places to put those mutations and different types of mutations. And we want to be able to summarize this information. So the idea we had in order to provide such a summary was to use this sort of graphical representation. So what we did is we moved along the phylogeny starting from the top to the bottom, and every time we encounter a mutation, then we draw a segment. Here we had a C and we moved to an A, so we have a C to A mutation, so we draw this arrow here vertically. Then we move to the bottom, and the next mutation was from an A to a T, so we draw a new segment here that goes here in another direction. So our idea was to summarize graphically those histories of mutations along the phylogeny. And the sort of results we get looks like that. So that looks a bit messy, but actually it's not that messy if you look at it carefully. So here the top of the tree, so the most ancient mutations are around here, and as you go towards the present, you move to the periphery of this diagram. And what's nice to observe here is that at the periphery we always observe this sort of mutations. And here we did that in the context of simulations where indeed those mutations here had a higher probability to occur near the tips. So we were able to actually visualize that those sort of mutations were preferred near the tips. And the last slide is the same technique but applied to HIV sequences. And what was nice here to observe is that we had sites where we had those kind of nasty potatoes where we can't tell much about the processes going on. But for other sites, we observed trajectories like that that seem way more constrained than the other trajectories here. So those sites are really interesting because the mutations that occur at these particular positions of HIV sequences are very much constrained by some sort of force. So it's probably the impact of natural selections that we are seeing here in those sort of diagrams. So we are starting to have some interesting results, and hopefully in the coming 2 or 3 months, we'll be able to really provide nice explanations as to why we observe these sort of trajectories.
So, thank you very much. So we have a short time for one question. I have one question actually, so, but Fabio.
Very quick question. Could you use this to distinguish between adaptive and negative selection? I mean, different types of selection.
So I haven't thought about this too deeply. What I'm sure about is that there's some sort of selection going on here on the right-hand side. I don't know if it's negative selection or if it's positive selection, but quite clearly what's happening on the right-hand side tells us that the standard Markov models that we're using to describe those sites, they're just completely rubbish.
Starting from such kind of analysis, it may be very naive, my question, but could you also tell, for example, biologists, if there are specific molecular toolbox that are more involved in this evolution than others? I mean, because for a biologist, of course, the gene means a lot, but also how genes work to some molecular mechanism, and all the protein toolbox, for example, that is involved would be also very valuable information to understand where evolution actually acts on, which object act in the cell.
Okay. So I'm not quite sure what the question is.
Okay, so I mean, there you identify things at the level of the genome,
yeah.
But then it would be also interesting to describe this, for example, at the level of the proteome, for example.
Yeah.
And because this would provide an element for further biological investigation.
So here, indeed, we're focusing on a very fine scale of genome evolution, but people have used this sort of analysis where they map mutations along a tree, and they've searched for correlations across mutation maps that were inferred from different genes, right? So you can look at different genes, find correlations, and that tells you about some level of interactions maybe between the proteins that were encoded.
Okay. Thank you. So thank you very much to all speakers, and then we have a pause, coffee pause, and we start in 15 minutes, I think, because we are a little bit late. Okay?
Donc on reprend, on fait une petite pause là de 15-20 minutes et puis on reprend pour les derniers exposés. Je vous rappelle qu'à la fin, il y aura aussi la remise des prix Vladimir LIRMM. Voilà. Donc à tout de suite.
J'étais un peu long.
T'en fous, toi?
Ça, c'est la clé USB. Ta présentation, je pense qu'elle était déjà... Je ne sais pas,